Hey guys, and welcome to our Exam 1 review for Bio 1406. I'm going to briefly walk through the study guide that I posted on Canvas as well as some supplemental stuff. So just a little side note, guys, this is not an exclusive review as to what is fair game for the exam, but it's more of a way to get your thoughts organized as well as help you with your notes and some study tips. So before I even get started, I want us to look at our Canvas page real quick. So if we go to our Canvas page, we can go to modules. And if you scroll to where I have exam 1, so if it wants to load, so exam 1 right here, so in between weeks 3 and 4, I updated the exam page. So I updated it just a little bit. I added 3 additional things. So I added this introduction to biological concepts. So here is what it looks like if you were to open it. So this is just a brief summary. It's not a comprehensive vocab list, but it is something to get your minds thinking as to what vocab I feel that is the most important. So I think it's only like 2 or 3 pages, nothing too crazy. And most of the stuff we have definitely talked about numerous times. And then the other thing that I added was this organic compound document. So it looks like this. So we kind of went over this in class this past week. So this is mainly for chapter 3. So like I said, this is kind of just a basic summary of like the big picture vocab words as well as some key concepts from that chapter. But just remember, guys, this is not 100%, okay? It's just to help you. And one last thing. So I know I did mention not to buy the textbook because it is uber expensive. So I found a online textbook that is completely free. It doesn't match up with our chapters exactly. So like chapter 1, I don't think goes over evolution. But what you can do though is you can go here to OpenStax. So this URL link. And this is what it looks like. So this is what it will pull up. So what you can do is pull up an online version. You can actually physically like type in a search bar like a concept or a vocab word, and it'll take you to that page if you want. And then you can also go into table of contents where you can look at the chapters and everything. So for the most part, it might be almost the same for the first 3 chapters, but I think as it progresses further on, it gets a little different. But this is something, an online textbook that you can definitely use that will match up with the same content as our textbook that is quote unquote required for our course. So this can definitely help you. This can even help you with the reading quizzes as well. So definitely I would say use this if you did not purchase the textbook, which is perfectly fine if you did not. So that's another resource for y'all, okay? So I'm giving y'all a lot of resources and I hope y'all utilize them. So let's go ahead and get started. So I'm just going to walk through the exam review. So the first question was, what are the 4 main points of Darwin's theory? So the 4 main points of Darwin's theory, so I'm going to go ahead and pull it up in our PowerPoint. So let me find the slide real quick, guys. So the 4 main points of Darwin's theory is generally this figure coming up. Sorry, guys. So these are the 4 main points. So all the population has variable traits. These traits have to be heritable, meaning that they have to be able to be passed on from parent to offspring. And then if there is overpopulation and resources become limited, such as food or area, then competition will occur. And then also the organisms that have that favorable trait for the environment, they will survive and their offsprings will obtain that trait. So this is the figure that shows that. So we have these beetles. So we have, looks like 2 different kinds of beetles. So we have white beetles and darker beetles. So if you put them on them in a black area, a bird comes along, predation occurs. The black beetles, the darker beetles, are going to be more camouflage as opposed to the white or lighter colored beetles. So with that, the darker colored beetles are going to start reproducing a lot more, and their offsprings are going to get their traits. And then with that, there is increased frequency, so increased occurrence of that favorable trait. So those are the 4 main points of Darwin's theory. Next question on the review is, what does it mean to have a hypothesis, a theory, and a law within science? So let me go ahead and type that out. So a hypothesis is a guess, so a logical guess. So let me correct that. So a logical guess with rationale, and you made that guess based on a observation. Based on an observation, okay? And then also with hypotheses, they have to be testable. They have to be testable, okay? So you have to be able to physically run an experiment with them, okay? So next, what is a theory? Sorry guys, I can't spell tonight. So what is a theory? So a theory is generally one or more hypotheses together. Is one or more hypotheses together, and they have been this theory, so it has been supported by repeatable testing. So meaning you ran the experiment countless times and you found the same result, just like Darwin. So an example is natural selection. So natural selection, Darwin saw this, so he tested it numerous times across multiple species. So the biggest species that he tested with was the finches, the tortoises, and the iguanas, the marine iguanas. So he tested that. He saw that numerous times that with his theory of evolution, he saw that across multiple species, and the fact that he ran tests, he made clear observations, and he saw consistent results, it formed a theory. Now, a law in science is a short, sweet statement that has not been proven wrong. Meaning it is always true. So it is always true. So an example of that, like I said in class, was thermodynamics. Sorry guys, like I said, I am not a good speller. So thermodynamics. So the first law of thermodynamics says energy is not created nor destroyed, only transferred. So that law of thermodynamics has never been disproven. It always remains true. So that is whenever it becomes a finite law. Now, with laws and theories, they can always be evolving. That does not mean they were necessarily wrong. It just means that new information was discovered. That's all. It doesn't mean that it's 100% wrong or anything, but based on the current information that they had at the time, it was true. And then also, as time goes on, things can always progress. So you can always add a new law of something, or you can always add another component of a theory. So just know that these are not 100% rigid. They definitely can develop and mature as time goes on. Okay, so now we're going to talk about the differences between independent and dependent variables. So independent variables, so I'm just going to abbreviate, guys. So independent variables, these are the variables that you change. So you change them. You change these variables during an experiment. Okay, so you change these guys during experiment. So let me actually zoom in a little bit so y'all can see this a little bit better. Sorry. So these are the variables that you change during experiment, such as like temperature or something. So like you change the temperature during an experiment. Whereas a dependent variable, this is something that is dependent on the independent variable. Sorry guys, I really can't spell tonight. I'm just gonna abbreviate. So it's dependent on the independent variables, such as, so let's say, so we learned a little bit about enzymes in class. So let's say that we increase the temperature, and with that, we denature our enzyme. We denature our enzyme. So the fact that we increase temperature, the enzyme is dependent on its structure and function because of temperature. So let's say, what if we decreased the temperature? The enzyme would not work, would not undergo a reaction. Okay? So it would not undergo a reaction, whereas if you increase the temperature too high, you denature the protein and no function. So enzyme, so no more function with the enzyme if it is denatured, okay? So definitely think about examples of independent, dependent variables. That is something that I might ask you on a test. And then the differences between a controlled and experimental group. So these are both groups that we have. The only difference between the two groups is that the independent variable is the one that is changed within the experimental group. So let me write that out for y'all. So the difference between controlled and experimental groups is that in the experimental group, you are changing the independent variable, okay? So that's the only thing that's difference between them. And then the independent variable in the independent variable is quote unquote controlled. So let me write this out. So the independent variable is controlled, which means, meaning it is kept constant. So you do not change it. So you never change it. You never change it throughout your experiment. So that's what it means whenever it is in the control group. Okay? Okay, so next, the difference between quantitative and qualitative. So the big difference, so let's go to our PowerPoint. So our PowerPoint, where you can find that. So the two different types of data is quantitative and qualitative. So qualitative, so remember that L means how does it look? And then quantitative, remember that N means that you have a lot of numbers. So that means that you record actual number data. And a lot of times you report that in a table or a graph of some sort. Excuse me. So definitely no differences between quantitative and qualitative, and some examples between them. Okay, and then also know that with qualitative, you can evolve it into a more quantitative data. So like, for example, we can say that our test subjects, they all have brown hair. To make it more quantitative, what you will do is that you'll be like, within our testing population, we have 5 that are brown-haired. So you can make it a little bit more quantitative by adding a number to it and actually recording that number. Okay, so before we wrap up the concepts of chapter 1, I definitely want you to look over the scientific method. So you have to make an observation. From that observation, you are thinking of a question. So like, why is that occurring? And then you come up with a hypothesis saying that something is happening because of XYZ. And then that is also kind of tied into your prediction. So if you were to run an experiment, you would expect a certain outcome to support your hypothesis. And then you would physically run your experiment. And then from your experiment, you will obtain results and analyze. And then based on your results, if your hypothesis was rejected, a lot of times you go back to the hypothesis place and then you rework your hypothesis and start over again. But if your results support your hypothesis, then a lot of times you become a theory, kind of like what happened with Darwin. So you'll become a theory, and then you'll start all over again if you're starting like a new project or something. So this is the idea of the scientific method. So definitely think of that while you are studying. Keep that in mind and think about possible like case studies for the scientific method. So if I was to give you a case study, I would want you to answer questions based on a short case study. Kind of like the one that I gave on the review. So I gave a little case study review right here. It's not the best, but it definitely gets your mind thinking as to like, what was the independent and dependent variable? What were they asking? So definitely look into that. If y'all want to make sure that you're studying the right thing, please feel free to email me with some answers and questions, and I'll try to help as much as I can. Okay, so now we're going to move into chapter 2. So chapter 2 was the basics of chemistry. So we are going to go over the question that says, what is matter, element, an atom, an isotope, ion, compound, and molecule? So let me actually, oopsies, sorry guys, let me find this document so I can copy and paste something real quick. So I just want to copy this table so we can talk about it. So, what is matter? So matter, the basic idea is that it takes up space and it has mass. So matter is made up of elements such as like hydrogen or carbon, nitrogen, all those bad boys that are found on the periodic table. So an element cannot be broken down. So you cannot go smaller than one atom of a hydrogen. There's nothing smaller than that. So it can't be broken down by a chemical reaction, and it's considered a pure substance. What is an atom? An atom is the smallest unit of matter. It has 3 main properties. So it has a proton, neutron, and electron. Protons, positively charged. Neutrons, no charge. And electron, negatively charged. And then what is an isotope? So an isotope is a different form of an element that has the same number of protons but different neutrons. And the fact that it has a different number of neutrons is why the mass is different, but the atomic number remains the same for the element. An ion, so this is a transfer of electrons, and both atoms will have a charge. Whether a charge is positive or negative is to be determined. So if it has a positive charge, it's considered a cation, so that little T looks like a plus sign. So if you know one, you know the other. So anion is negative. And then also just a little side note, guys, whenever you're studying biology, it's good to learn things in pairs, because a lot of times if you know one, you know the other, because they're a lot of times the opposite of each other. So like right here, cation, anion, if you know one, 9 times out of 10, you know the other one. So just a little side note. A compound is a molecule that is made up of 2 or more elements in a specific ratio, such as sugar. So sugar is C6H12O6. So this is in a specific ratio, and it will never happen outside of this ratio within a compound. And a molecule is a group of atoms that are bonded together, and they also represent a fundamental unit of a chemical compound. And a lot of times these molecules happen whenever a chemical reaction takes place. So that is the basic idea of those guys. Next, what we're going to talk about is protons, neutrons, and electrons. So let me just enter down. So I'm going to start writing for y'all guys. So let me just get my pen going. Okay, so we're going to talk about a proton, neutron, and electron. So protons, they're positive. Yay, positive. Neutrons, 0. They have no charge. So this is in the context of charge, so let me write that. And then electrons are negative. These are the mopey bopes. Sad face. Okay, now where are these guys located? So where's their location? Sorry guys, my handwriting is not that good on this. Location, so protons and neutrons are found in the nucleus. And I spelled nucleus wrong. Is found in the nucleus. Same thing for neutrons. And then electrons are found in the cloud. So they surround the nucleus. Okay? So they look something like this. So let's do, how about we just do carbon, because carbon's super easy. So carbon 12, I'm sorry, carbon 6. So we're gonna do carbon. So let's make his nucleus. Let me actually make that a little bit bigger. So let's pull up a periodic table. Okay, so carbon, his atomic number is 6, as we see right here. So carbon right here, he is number 6. So carbon 6, that means that he has 6 protons. Neutrons, we're going to find out. So he has number of protons equals 6. His mass, let's look at their periodic table again, guys. His mass is 12. So as we see right there, carbon has a mass of 12. If it wants to load. So his standard mass is 12. So then from that, how do we find number of neutrons? You can find that formula within our chapter 2 PowerPoint. So right here, in order to find mass number is the number of protons plus the number of neutrons. So let's rearrange that a little bit. So, oopsies. So mass number equals so mass equals number of protons plus the number of neutrons. So let's solve for neutrons. So let's solve for this guy. So mass is 12. Protons is 6. That means neutrons equals 6. So we have 6 neutrons as well. Okay, so now what we're going to do is figure out our electrons. So electrons are always the same number of protons. So we have 6 electrons. So we're going to create our first shell. So let me change colors here, guys. So let's put a red. So on our first shell, we always have 2. And then on our second shell, we can hold up to 8 after that first shell on every one. So we have 1, 2, 3, 4, 5, and 6. So how many valence electrons does he have? So valence electrons. So how many electrons does he have on the outermost shell? He has 1, 2, 3, and 4. So he has 4 valence electrons. So I will expect you to be able to do this on the test. I may not give you carbon, I might give you something else, maybe like calcium, anything of that nature. So definitely be prepared for that, okay guys? So I just walked through an example, we should be good. If you have questions though, definitely email. Don't hesitate to ask, okay? Okie dokie. So now we're gonna go over the different types of bonding. So we have polar, covalent, nonpolar, covalent, and what was the last one? Ionic bonding. So let me pull up one table. So let me pull up this table. Oopsies. Sorry guys, technical difficulties. Okay, so now let's look at this table. So we have covalent. So covalent bonding is the equal sharing of atoms. So this can occur between multiple atoms. It can be, it has to be 2 or more. And what happens is that whenever 2 or more atoms, they're held together by a covalent bond, that makes a molecule. Also, double bonds can occur. So we'll go over some examples of that in a moment. But polar covalent is a type of chemical bond where the electrons are unequally shared, meaning that there's a slight pull to a certain atom, such as water. So let me type that in. So water is the perfect example of a polar covalent, if I can spell. It's a perfect example of polar covalent. So I'll show you a picture of that. Nonpolar covalent is a bond between 2 hydrogen atoms, and they equally share the same electrons. So another example of that can be like 2 chlorine atoms. They are equally sharing their electrons. So kind of very similar to covalent bond, but nonpolar is a little bit more specific as to that it will occur between 2 of the same atoms, whereas covalent bond will occur between different kinds of atoms. And then ionic bonding, so this is whenever we have the stripping of valence electrons between atoms. This bond can occur between cations and anions, so it will produce cations and anions. And generally you'll form like a salt, such as NaCl, so table salt. So let's go to our PowerPoint and let's find that information. Okay, so a covalent bond, so like I said, this is an example of a polar covalent bond, this image right here. So this is a non-polar covalent bond. So hydrogen has 2 electrons. So generally they each have 1 valence electrons just floating around. And then what will happen is that they will come together and they'll form a covalent bond with each other. So this is a non-polar because they are equally sharing it. There's no slight pull. And then more examples of covalent bonds. So water, like I said, is an example of a polar one. And the reason why is because the electrons from the hydrogen, so the electrons right here from the hydrogen, they slightly get pulled towards that oxygen molecule. So there's a little bit of pullage between them. Not much, but just enough to where they're not 100% always equally shared. Now with oxygen, as I said, that there could be double bonds, so let me zoom in. So there could be some double bonds. So this is an example of just a single bond, so the hydrogen to hydrogen. Whereas oxygen, they will share a total of 4 valence electrons in the center, and then each pair of electrons, so each pair of electron makes a bond. So it's kind of like this, guys. So there's a bond in between, there's two bonds in between the oxygen molecules. So just know that it's not always just one bond, it can go into multiples. Okay, so now let's go, let me find another example of a polar one. So as you see right here, so like I said before, like water, like there is so hydrogen is generally positive and oxygen is generally negative. So the fact that oxygen is generally negative, he prefers, so he has higher electronegativity, meaning that he attracts electrons. He has a higher affinity for them. So with that, that's the reason why there is this pullage going towards the oxygen, and that is why it's a polar covalent bond, because of that slight pull of electrons, because he is more electronegative. Okay, and then ionic bonds, the last one that we're going to talk about. So right here in this figure, as I said, so sodium has one valence electron, as you see in red. So that one valence electron, what happens is that, like I said in class, it's easier to just get rid of one than it is to gain 7 additional electrons around him. Whereas in the fact of chlorine, chlorine has 7 electrons, so it's easier for him just to gain one. So what will happen is that sodium will donate his electron to chlorine, so they are both happy. Whereas you see in the final result, sodium has 8 valence electrons on his outer shell, and so does chlorine. So that is what they always want. Everybody wants to be happy. Everybody wants to have 8 friends. So just know that if you lose an electron, you are a cation. If you gain an electron, you are an anion. And the reason why is because negatively charged attracts negative. So like attracts like in this case. Okay, so next concept on our list is hydrogen bonding. So hydrogen bonding, sorry guys. Hydrogen bonding, they're extremely important. So they are generally weak. So by themselves, hydrogen bond is very weak, but collectively they are very strong. And then an advantageous trait for this is with water. So with that, we're going to transition into water. Like, why is water important? So let's go to that. So we have 4 emergent properties, so definitely know what an emergent property means, guys. So it has cohesive ability as well as adhesive ability. So cohesive ability or cohesive behavior is whenever it is sticking to self. So water sticks to water. And then adhesion, so let me type that in for y'all. So adhesion behavior, adhesion behavior is whenever it sticks to other molecules. So that's the difference between cohesion. So co means same, so like together, like co, together. Adhesion, you add a different molecule into water. Another thing is that it has the ability to regulate temperature. So us personally as humans, we use this evaporating system. So with water being able to be evaporated, heat is being released, and then also with heat being released, we are cooling ourselves. And then also the expansion during freezing, so this is why water floats. So with the fact that it forms this very uniform structure, that is why we don't see ice sink, because ice is less dense. And then also, if ice did sink, all water on Earth would just eventually freeze over, and then we'll just be in another ice age and life would not be possible. And then lastly, water is a universal solvent. Meaning, so with it being a good solvent, that means that it's very good at dissolving a lot of ion. So it's good at dissolving salt. It's good at dissolving other polar molecules. But it is very bad at dissolving nonpolar molecules. So, such as oil. That's why oil sits on top of water because water cannot dissolve it. And then these unique properties, why does it have all these unique properties? It has everything to do with how it is bonded. So the fact that it has a polar covalent bond is the reason why it has um, all of these unique properties. So with that, I'm gonna say the F word. It's fabulous. So water is absolutely fabulous. Okay, so one last thing for this chapter is the pH scale and acids and bases. So if you are acidic, you are roughly 1 to 6 in range. Water is considered neutral, so the 7, and then 8 to 14 is basic. So for that, just know what the scale is, the pH scale, and then also know that with acids, you have a higher concentration of hydrogens as compared to bases, and bases have a higher concentration of OHs as compared to acids. So that's the main difference between them, between acids and bases, okay? Okay, so that will conclude chapter 2. Now we're going to transition into chapter 3 about our carbon and organic compounds. So let me pull up chapter 3. Okay, so why is carbon so fabulous? So carbon is absolutely fabulous because he has 4 valence electrons, meaning that he has the potential of forming 4 covalent bonds. And with that is what makes him so versatile. And with the fact that he has 4 various opportunities, he can create multiple shapes of himself, whether that be in just straight chains, such as like fatty acid chains, or be in like ring structures for like hormones or something of that nature. So carbon, extremely versatile. We love carbon. Okay. So now we're going to transition into functional groups. So in the review, I did provide a basic table. Of those, I feel that they are probably like the most important, as well as the aldehyde group. So let me pull up the OneNote real quick and let me start drawing. So an aldehyde, so it's really hard to see in that picture, but an aldehyde is a carbon double bonded to an oxygen and singly bonded to a hydrogen. So this is an aldehyde. And I can't spell. So this is an aldehyde. So definitely know this guy. He is extremely important as well, in addition to the ones that I did provide. So let's just go ahead and go through that table. So we have a hydroxyl group, an OH. Please recognize OH. It is not HO. It is not this. There are no H's in biology, okay? No H's, okay? It has to be OH. This is correct. So on a test, if I were to put HO instead of OH, please do not select the Ho. No Ho's, okay? So only OH's. So OH, like I said, is a hydroxyl. And where you can find these guys, they are mainly found in carbs. Let me actually put this in a nice little table, guys. It's getting a little sloppy. So we have hydroxyl, so this is what he looks like. So like I said, his name is hydroxyl, and where you can find him is generally in carbs and then in alcohol. Those are the 2 main places where you will find him biologically. Next group is a carboxyl. Excuse me. This is a carboxyl, and this is a carbon double bonded oxygen to an OH. You may also see him written as this. That is the condensed way of writing it. You can find him in two main places. You can find him in fatty acids and proteins. Okay, next we are going to talk about aminos. So amino is a nitrogen with two hydrogens, and these guys are primarily found in proteins or amino acids. So same thing. So amino acids is the monomer of proteins. Okay. And then two more. So we have the sulfhydryl, can't even say it. So this is the S, the SH. So he has sulfur, amino. So actually I'll go back and I'll talk about that a little bit later. So we have the sulfur group. He is found in amino acids. Okay, and lastly is the phosphate. So phosphate, there's numerous ways to write him. I'm going to draw him out. So actually I'm going to draw him in the abbreviated form. So this is the abbreviated form of phosphate. But you may also see him like this. So let me pull up in our PowerPoint. You may also see him like this, guys. So right here, you may also see him like this. Okay, so definitely realize that it's a phosphate group. He's the only group with the P in it. Okay, so phosphate, he is a huge player. He is everywhere. ATP. He's in nucleic acids. And he's also in our phospholipid, in our membrane. So phosphate, huge. Like, you are going to be sick and tired of phosphates by the time you are done with this course. So an easy way to remember these guys, the three easiest ones to remember. Amino, there's an N, there's a nitrogen. Sulfur, sulfhydryl, sorry guys, I can't say it. This guy, he has sulfur, he has an S in it. And lastly, phosphate, P. Those are the three easiest ways to remember at least these guys. The other ones, create your own little mnemonics or anything like that, anything that works for your brain. Okay. Now, we're going to talk about what is the reaction that happens in order to build up these macromolecules that we talk about. So the one to build up a macromolecule, it is known as dehydration reaction. So if you are dehydrating, you are taking away water. Water goes away. So a hydrogen and an OH group from two separate molecules, they will come together and they will pop off and make water, and then they will create a longer polymer. They will create a new polymer. Okay, so that's dehydration. Now what's the opposite of dehydration? Is hydrolysis, meaning that you're breaking apart water. So water is HOH written out rather than H2O. So these are the same thing, they mean the same thing. So what happens is that you break this water molecule right here. hydrogen will go to one molecule, and then the OH will go to another molecule. So that's why it's called hydrolysis, because you are lysing, so breaking a water molecule. And with that, what you're doing is that you're breaking apart that longer polymer that we just made into two shorter chains. Okay, and the molecule that is involved in both of these reactions is water. Water, 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 okay? Okay, so next question, a lipid. So why is a lipid not a polymer? So a lipid, so let me go to that slide and let me start talking about it if I can find it. So lipids are not true polymers, and the reason why is because they are not made up of repeating subunits, meaning they are not made up of the same monomer, kind of like carbohydrates. So carbohydrates are repeatedly made with multiple sugar molecules, whereas lipids are not. The smaller molecules that make up a fat, so let me go to that slide. So the molecules that make up a fat is a glycerol and fatty acid chains. So a glycerol, I'm going to draw that out so it's a little bit bigger for you guys. So a glycerol is a 3-carbon chain with some hydroxyl groups on there. And then we have some hydrogens over here. So this is a glycerol. Okay, and then what will happen is that we will have a fatty acid chain, so we'll literally just have a long chain of carbons. And then on one of the ends over here, what will happen is that you have a carboxylic acid on a fatty acid chain. So you have this carboxylic acid right here. And what will happen is that this glycerol will come in contact with this long carbon chain. And then what will happen is that this OH and this hydrogen from each glycerol, so you'll have 3 of them, so you'll have multiples. It'll happen 3 times. And then this hydrogen will pop off. And then with those popping off, we get water. Yay! Sorry guys, that's a really ugly water. We get water. Yay! When in doubt, water, guys. So, you'll get water. So, the 2 molecules that make up a fat is a glycerol and a fatty acid. And then once these guys come together, I'm going to zoom in, we get an ester linkage. So, an ester linkage looks like this. And then you have your fatty acid chain, and then you have your glycerol backbone. Okay, so right here is another functional group, guys, that you need to know. It is called an ester. And then these are specific to fats, okay? One more thing. Actually, 2 more things, just kidding. We have 2 different types of fatty acids. So we have saturated and unsaturated. So saturated, they have all of these single bonds like they're just straight lines. And they're solid at room temperature like butter. And then we have unsaturated, which is where there's a double bond that occurs. So let me zoom in on that. So there's this double bond right here. And with that, it causes the fatty acid chain to kind of curve. So that's called a kink. Okay? So kinks are very important. And we'll get more into why kinks are important whenever we get into cellular membranes. But just know that in the context for this slide, an unsaturated fat has a kink in its chain, meaning that there is a double bond present. And then an example of that are oils. So that's an example. And then one last thing is phospholipids. So phospholipids are extremely important, especially whenever it comes to our cellular membrane. So a phospholipid has a hydrophilic, so water-loving head. So water-loving head right here. So this is the head. And then we have a hydrophobic tails, which are these guys. So a phospholipid, you will have one straight leg, and then you'll have a kinky leg. So they're dancing. Think of dancing legs, okay? So definitely know those. Know the components of a head. So the head is comprised of a glycerol and a phosphate. The chlorine I'm not so interested in. The big picture though, guys, I really want you to know that there's a phosphate group as well as a glycerol. So the glycerol is what actually links the phosphate indirectly to the fatty acid. Excuse me, the fatty acid tails, okay? And where can we find phospholipids? Our cellular membrane. Okay, steroids. So steroids are cool. We'll go more into little detail later on. You'll also probably hear about it in anatomy and physiology if you take those courses. But for steroids, I want you to know that it is made up of a 4-ringed structure that is comprised of carbon. And the biggest steroids that I want you to know are cholesterol and some sex hormones. So like progesterone, estrogen, what's another one? Estrogen, progesterone, and testosterone. There we go. Cholesterol, definitely know why cholesterol is important. So cholesterol is found within our membrane. So let me go back to this picture. So this is an example of our membrane right here. Randomly throughout our membrane, you will get these little cholesterols in between these little, in between the phospholipids. You'll get cholesterol just randomly throughout. And the importance of that is because it creates fluidity. So our membranes are very malleable in a sense where they're like waves. They're constantly just flowing, going with the flow. So definitely know that. A steroid generally will look something like this. So actually let me go into our other one. No, it'll be easier to draw in there. So a steroid, whoopsies. So a steroid, so it's generally a 4-ring structure. Oops, if I can draw. And then the 4th ring though is, so you'll have generally 3 rings that have 6 carbons, so these 3, and then the last one will only be a 5-ring. So that's the general basics of the backbone of a steroid. Almost all steroids will have this backbone, if not all, and then what will vary is what branches off of these rings. I won't expect you to know that, but I do want you to know the general backbone of a steroid. Okie dokie. So now we're going to go into our macromolecules more specifically. So let me go back to our PowerPoint. So we're going to start off with carbs. So carbs, the monomer of a carb is a monosaccharide. So this guy, monosaccharide. And then the polymer of it is a disaccharide, so multiple sugar links like it shows inside at the bottom. So that's a polysaccharide because it's more than one. A disaccharide is just 2, so if this picture just cut off right here, so just cut off right here, these are disaccharides now. And the link that happens between them, as you see in the green circle, that's a glycosidic linkage. So it's specific, so it looks with an oxygen with sugars on either side of it. Okay? And then the function of a carbohydrate is chemical energy and ATP. So since I mentioned ATP again, I do want you to know what ATP generally looks like. So we'll have adenosine, and then you'll have 3 phosphate groups. Okay? And then with ATP, the way that we get energy from ATP is by cleaving, meaning that we pop off a phosphate group. So he goes away, and by doing that, we add water. So we do add water, and then it pops it off, and then with that, it produces energy. So now we have a phosphate group just floating around that can bond to anything else that's needed. And then we have a burst of energy. So this will happen constantly throughout our whole body without us even knowing it. Okay, so next we're going to talk about proteins. So let me scroll down and go to proteins. So proteins, so like I said in class today, there was a lot of structures and functions that go with proteins. So there are 8 various functions that proteins do. We will go more into detail into their functions as we progress throughout term, but as of right now, I just want you to know the big picture. So just know that enzymatic reactions, it helps produce antibodies, it helps with cellular transport, it also helps with muscle movement, and specifically with muscle movement, it talks about actin and myosin. So these are 2 protein structures that are along our muscle fibers, and they actually contract and everything that help our muscle move. So those are just some big ones. So a general structure of a protein is that it will have this amino group, meaning it has this the NH2 group. And then it will have a central carbon in the middle of some sort, or somewhere along the chain. And then on the other end, at the very opposite end, so this carbon chain can vary. It doesn't have to just be 1. It can, I think it could go up to 2 or something in the center. But you'll have a alpha carbon, and then you will have this carboxyl group, which is that COO group, COOH group. And then this R group, so this R group is the one that's variable. So this is the one that can produce like many different structures, some of them as simple as just 1 additional carbon, or even as complex as a ring structure. So that will vary. And then with that, that's why we can get so many different amino acids. And we have a total of 20 amino acids within our body that we do synthesize. And then in order to make an amino acid chain, we have to have 2 amino acids. So these are 2 separate amino acids. So this is 1 amino acid, and this is another amino acid. So what will happen is that on one end, we'll have the amino group actually on one of them will interact with the carboxyl group on the other side. And then whenever that happens, I wonder if I can change my color, my pen color. Oh my goodness, I can, guys. I didn't know I can do that in PowerPoint. So let me change color. Let's do blue. Blue's fun. So what will happen is that the OH group from the carboxyl group and the hydrogen group from the amino group will pop off and produce water. Oh my goodness, it's water again, guys. So that means he's important. I've said him at least 20 times. Water plays a vital role. And with that, with an OH and a hydrogen popped away, the carbon and nitrogen will then bond as you see below. And this bond between the carbon and the nitrogen is called a peptide bond. So there can be numerous peptide bonds. It can range from just 1 or up to thousands, like hundreds, thousands. It just depends. So just know that in order to get a polypeptide, so many peptide bonds, there has to be that dehydration reaction has to occur, okay? And then with that, the function of them, so overall function and structure, oopsies, wrong slide, sorry guys. So function, there are many functions like I said. So they come in the form of enzymes, they help in reactions, so they help in speeding it up, making it quicker. So rather the reaction taking 10 minutes, if an enzyme's added, it may only take 3 minutes. Like I said, with muscle movement, so we have actin, so we actively have actin, And then we have myosin. So these are the two proteins that play a big role in muscle movement as well as defenses. So they help make antibodies, and then also with transport. So they are found throughout our membrane. So whenever we get into cellular membranes, I'll go more into detail about those proteins, but they do help with transporting things from in and out of the cell. Okay. Now, the overall structure of a protein will vary. So almost all proteins will have the first three levels. So they will all have primary, secondary, and tertiary. Now, whether or not they go into the quaternary structure will vary, but the primary function of these guys is to make proteins. So in order for you to have good cellular function and everything, you have to synthesize proteins, and structure will dictate function. So with primary, it's literally just a string. So if you think of a shoelace, so if you have like a shoelace, and then you just have one long string, and then you want to go to the secondary, so what you'll do is start twisting it. You can twist it, and then you can get that alpha helix, or if you fold that shoestring, you can get beta pleats. And then what will happen is that, so in order to get a secondary structure, there has to be a reaction within hydrogen bonding. So hydrogen bondings are occurring. And then what will happen with the tertiary structure, in order to get a tertiary structure of the alpha helix and the beta pleats working together, there is an interaction between that R group that I talked about. So that R group is what the interactions between them will dictate how the tertiary structure will work. And then as well as, I didn't mention this in class, but I'm probably not going to ask you, but just a little side note. So I wrote right here, so there's disulfide bridges. So disulfide bridges means that there's sulfur present. So there are some sulfur bonds that are also happening between here. So just a little side note, guys. And then quaternary structure. Not all proteins will form a quaternary structure. They are extremely complex, as you see. Like, this can just be, to me it looks like a hot mess, and they, in real life, they do look like hot messes, but they are extremely important. So a lot of times the way I think of it is that if it has the higher form of structure, it has greater function. So an example of like quaternary structure is like hemoglobin. Hemoglobin is huge for transporting oxygen within our blood. If we did not have hemoglobin, I don't know how we would survive. As well as DNA polymerase. So DNA polymerase plays a huge role in replicating and translating and transcribing our genomic information, so that's huge. And then as well as ion channels. So quaternary structures within the cellular membrane, they help transport ions throughout, in and out of our cells, such as like sodium, potassium, calcium. It's huge. So it's very, very important. Okay, so now we're going to go into nucleic acids. So definitely know the components of a nucleotide. So components of a nucleotide, there are three main ones. Excuse me. There is the phosphate group, the sugar base, and then the nitrogenous base. So the nitrogenous bases, there are 5. One of them is strictly RNA based, and one of them is strictly DNA based. So definitely know the differences. So know that cytosine, thymine, adenine, guanine, these are all found in DNA, whereas cytosine, uracil, adenine, and guanine, they are all RNA based. As well as know the differences on how they are separated, meaning that pyrimidines are single ringed and purines are double ringed. Okay? So now, depending on if you are making DNA or making RNA will determine what sugar base you will have. So if you are making DNA, you are going to have a deoxyribose base. And if you are making RNA, you will have the ribose sugar base. Now the little side chain over here on whether or not if these nitrogenous bases are going to be added just matters on coding system. So if we need an A, we'll add an A to it. So the main difference between DNA and RNA is summarized in this picture. So DNA is double-stranded, RNA is single. DNA has, like I said, deoxyribose. So the main difference between the deoxyribose and just ribose is that RNA has this additional hydroxyl group right here. So if I were to give you a picture and be like, what is the difference between them? Or if I was to be like, this sugar base, is it DNA or is it an RNA base, sugar base? So you would have to look down here and be like, okay, if there is two OHs, it goes to RNA. Okay, so definitely know that. As well as overall function for these guys. So let me go to that final table. So the overall function for DNA is to create and store genomic information, and then RNA is specifically the one that copies it, and then from that you can get protein synthesis. And we'll talk more about DNA and RNA later on in transcription and translation. And then where DNA can be found is in the nucleus. Nucleus only. That is the only place where you will ever find our DNA in terms of eukaryotes. And then RNA is originally made in the nucleus, and then it does have the potential of leaving the nucleus, but it does not leave the cell. So it stays within the cell and it just floats around in the cytoplasm. Okay. So one last thing on the review, I did see that I put a table that had some prefix and suffixes. So I'm just going to briefly go over those. So I'm going to write them real quick. So if you see something that says hydro, it means water. So it means water. If you see something that says lyse in the word, it means to break. And then philic is loving, so anything loving. So like hydrophilic, so water loving. And then we have GLY, so this means sugar. So it means that there's going to be carbon present. And then we have mono. So mono means one. Tri, which means three. Phobic, which means fearing. So hydrophobic, water fearing, meaning it doesn't like it. Poly, which means many. And then saccharid. Sorry guys, my handwriting is trash on my computer. Is sugar. And then if a word ends in ose, it also means sugar. So such as, goodness, I can't spell. Such as glucose. Oh my goodness. Okay, I'm gonna spell sucrose because I know how to spell sucrose. Sucrose. Oh my gosh, I'm so sorry guys. It's really late. You know what? Fructose. I can spell fructose. So fructose, so since it ends in ose, it means that it's a sugar. One last one, it's not on there, but di. So di, it means two. Okay guys, one last thing I am going to show y'all before I conclude this. So I'm going to scroll down real quick. I'm going to pull up this picture. I'm going to paste this real quick. Okay, so this guy, So these are some examples of structures that I may put on your test. So definitely, if you look at the molecule as a whole, be able to identify, is it a sugar, is it a fatty acid or amino acid or a nucleic acid? So definitely know that, guys. That's partly why I had you do the foldables. So be able to, like, if I were to like circle or color code, be like, what is this? Like, what is that functional group? Be like, that's a hydroxyl. So be able to do that throughout for the exam, okay? Um, so these are actually the answers for the exam review if you wanted to, um, for that portion of it, if you wanted to look at it. But just know that I may change it up a little bit. I may put, instead of like, this is actually a glucose molecule, I might put a sucrose molecule or something of that nature. But you should still be able to identify between them. Okay, so I hope this helped, guys. I really do. Let me just make another little disclaimer. This is not an exclusive, these are the only questions I'm going to ask or anything. It was just something to help you out, to get your thoughts motivated, to get you on the concepts that I feel that are like the most important, and then also to get your brain thinking as to how can she create that concept into a question. So definitely think about that. Definitely look at the supplemental stuff that I gave you. If you have any questions, don't hesitate to email. If there's nothing else, guys, I hope y'all have a fabulous weekend. Don't overstudy. Get rest, okay? If y'all have questions, please feel free to email me. And I think that wraps up our review for our first exam. So happy studying. Have a fabulous weekend, and I will see y'all on Monday.